Good day, this is Professor Will, CBMD PhD, today Saturday, May 9th, 2020. It is 1:53 p.m. Eastern Standard Time, and this is video 2 of the day on Corona series. So let's get started here. Timing of immune response to COVID-19 may contribute to disease severity. The interaction between the innate and the adaptive immune responses to COVID-19 may be impacting disease progression. Date May 1st, 2020. Source Keck School of Medicine of USC. Summary. A new study suggests that temporarily suppressing the body's immune system during the early stage of COVID-19 could help a patient avoid severe symptoms. That is, because the research shows that an interaction between the body's two main lines of defense may be causing the immune system to go into overdrive in some patients. The body's first line of defense, the innate immune response, starts right after an infection, like an infantry going after a foreign invader, killing the virus and any cells damaged by it. The second line of defense, the adaptive immune response, kicks in days later, if any virus remains, employing what it has learned about the virus to mobilize a variety of special forces such as T cells and B cells. Using the target cell limited model, a common mathematical model developed to understand the dynamics of viral infections, the researchers examined how the two immune responses work in COVID-19 patients compared to patients who have the flu. The flu is a fast-moving infection that attacks certain target cells on the surface of the upper respiratory system and kills almost all of the target cells within 2 to 3 days. The death of these cells deprives the virus of more targets to infect and allows the innate immune response time to clear the body of almost all of the virus before the adaptive system comes into play. Adaptive immune response kicking in too soon. But COVID-19, which targets surface cells throughout the respiratory system, including in the lungs, has an average incubation of 6 days and a much slower disease progression. Mathematical modeling suggests that the adaptive immune response may kick in before the target cells are depleted, slowing down the infection and interfering with the innate immune response's ability to kill off most of the virus quickly. The danger is, as the infection keeps going on, it will mobilize the whole of the adaptive immune response with its multiple layers. This longer duration of viral activity may lead to an overreaction of the immune system called a cytokine storm, which kills healthy cells, causing tissue damage. The interaction of the innate and adaptive immune responses might also explain why some COVID-19 patients experience 2 waves of the disease, appearing to get better before eventually getting much worse. Some COVID-19 patients may experience a resurgence of the disease after an apparent easing of symptoms. It is possible that the combined effect of the adaptive and the innate immune responses may reduce the virus to a low level temporarily. However, if the virus is not completely cleared and the target cells regenerate, the virus can take hold again and reach another peak. Counterintuitive treatment. The most provocative result of this research is the kind of treatment it suggests to prevent this interaction between the two immune responses. Based on the results of the mathematical modeling, we propose a counterintuitive idea that a short regimen of a proper immunosuppressant drug applied early in the disease process may improve a patient's outcome. With the right suppressive agent, researchers may be able to delay the adaptive immune response and prevent it from interfering with the innate immune response, which enables faster elimination of the virus and the infected cells. Small studies out of China, including a recent one of COVID-19 patients and one of SARS patients in 2003, show patients who received immunosuppressants such as corticosteroids had better results than those who did not. The researchers said a possible next step could be to take daily measurements of viral loads and other biomarkers in COVID-19 patients to see if the data validates the mathematical modeling. More preclinical studies, including experiments in animal models, will also be needed to prove the efficiency of an early immune suppressing treatment. Flatten coronavirus. What does it mean to flatten the curve when we talk about coronavirus? To limit the symptoms of the virus before we are too sick. To limit the rate at which the infection spreads. To speed up the time it takes to develop a vaccine. Coronavirus, SARS-CoV-2 infects cells of the intestine. Date May 4, 2020. Source Hubrecht Institute. Summary: researchers have found that the coronavirus SARS-CoV-2, which causes COVID-19, can infect the cells of the intestine and multiply there. Using state-of-the-art cell culture models of the human intestine, the researchers have successfully propagated the virus in vitro and monitored the response of the cells to the virus, providing a new cell culture model for the study of COVID-19. These findings can explain the observation that approximately 1/3 of COVID-19 patients experience gastrointestinal symptoms such as diarrhea, and the fact that the virus often can be detected in stool samples. Patients with COVID-19 show a variety of symptoms associated with respiratory organs, such as coughing, sneezing, shortness of breath, and fever. And the disease is transmitted via tiny droplets that are spread mainly through coughing and sneezing. 1/3 of the patients, however, also have gastrointestinal symptoms, such as nausea and diarrhea. In addition, the virus can be detected in human stool long after the respiratory symptoms have been resolved. This suggests that the virus can also spread via so-called fecal-oral transmission. Though the respiratory and gastrointestinal organs may seem very different, there are some key similarities. A particularly interesting similarity is the presence of the ACE2 receptor, the receptor through which the COVID-19 causing SARS-CoV-2 virus can enter the cells. The inside of the intestine is loaded with ACE2 receptors. However, until now it was unknown whether intestinal cells could actually get infected and produce virus particles. Researchers set out to determine whether the SARS-CoV-2 virus can directly infect the cells of the intestine, and if so, whether it can replicate there as well. They use human intestinal organoids, tiny versions of the human intestine that can be grown in the lab. These organoids contain the cells of the human intestinal lining, making them a compelling model to investigate infection by SARS-CoV-2. Infection of intestinal cells. When the researchers added the virus to the organoids, they were rapidly infected. The virus enters a subset of the virus in the intestinal organoids, and the number of cells that are infected increases over time. Using electron microscopy, an advanced way to visualize different components of the cell in great detail, the researchers found virus particles inside and outside the cells of the organoids. The researchers investigated the response of the intestinal cells to the virus with RNA sequencing, a method to the study of which genes are active in the cell. This revealed that the so-called interferon-stimulated genes are activated. These genes are known to combat viral infection. Future work will focus on these genes more carefully, and on how they could be used to develop new treatments. The researchers also cultured the organoids in different conditions that result in cells with higher and lower levels of ACE2 receptor, through which SARS-CoV-2 can enter the cells. To their surprise, they found that the virus infected cells with both high and low levels of the ACE2 receptor. Ultimately, these studies may lead to new ways to block the entry of the virus into our cells. The observations made in this study provide definite proof that SARS-CoV-2 can multiply in cells of the gastrointestinal tract. However, we don't yet know whether SARS-CoV-2 present in the intestines of COVID-19 patients plays a significant role in transmission. The current study is in line with other recent studies that identify gastrointestinal symptoms in a large fraction of COVID-19 patients and virus in the stool of patients free of respiratory symptoms. Special attention may be needed for those patients with gastrointestinal symptoms. More extensive testing using not only nose and throat swabs, but also rectal swabs or stool samples may thus be needed. Good day, this is Professor Will, CBMD PhD on infectious disease and microbiology. I hope you enjoyed listening to video 2 of the day, and have a great day.